Hey, everybody, this is Jason Wilson. I'm here with Dr. Anthony Smith, and we are with the Institute of Cannabinoid Medicine. And we are here today to talk to you all about the cannabis plant and cannabis products from a biochemist's perspective. So thanks so much, Anthony, for joining me for this little exploration. Hopefully we hit some good topics and teach people a few things.
Thank you, Jason. Looking forward to it.
The way we're going to break this down is kind of hit 3 or 4 primary points. To start, we're going to look at the cannabis plant and think about what are the chemical constituents of the plant, how do they vary, and then from there, we'll launch into a discussion about different types of cannabis and cannabinoid products and how their chemistry can differ between all of those products. And then we're going to lead into a discussion about how the different ways that we consume those products can affect us differently, and some of the science behind all of that, and see what else cannabinoid science we pepper in there. But to briefly summarize our backgrounds, Anthony, do you mind just sharing a little bit about some of the work that you and I have done together, some of the work that you've done that kind of has brought us to this point, why we are even studying the cannabis plant, and then we'll maybe launch from there to talk about things we've seen in the lab and see where it takes us.
Absolutely. My interest in cannabis stems from a lifelong interest in human physiology and human performance. I have a background in molecular biology and always been passionate about herbal and natural medicines. From a medical and health perspective, but certainly as an analytical chemist and biochemist as well. And in the plant world, certainly cannabis is very, very unique because it contains a vast library of different types of chemicals and compounds. And even within those specific types, cannabis has the genes to produce a myriad of compounds that are super interesting together and apart. And aside from its interest to humans as an entheogen and as a a sort of mystical compound, it's migrated into modern culture as a recreational drug, if you will. But its interest to scientists and chemists goes back, at least since the 1940s. And its lore, I think, in a medical context, is certainly in a great period of modern revival. But it's not a new concept, and that there's a lot that we do know about cannabis, especially from an analytical and maybe even safety perspective. But there's a lot that we don't know aside from anecdotal use and traditional cultural use about what it actually is as a medicine and what it can do and what it may or may not do.
Yeah, I mean, there's a lot of information around the mechanisms of action that are still being teased apart, but the sort of anecdotal record of medical cannabis use, definitely extends at least until the roughly around 5,000 years ago or so, and that's as far back as we can actually trace it. Probably goes further back than that. And the work that we've done in the past, we both kind of come from these laboratory backgrounds, analyzing plants in labs to try to see what's in them or how pure they are, that sort of thing. One thing that I wanted to bring up is this idea that when we talk about cannabis, we're not talking about one thing. Like, cannabis is not just cannabis. And some people kind of know that kind of intuitively, but maybe haven't really wrap their heads around just how diverse cannabis can be. And other people just maybe haven't even thought about that idea at all. And I hope by the end of this discussion, as we talk about the chemistry of the plant and all these differences, that'll be clear. But what comes to your mind when you think about, you know, cannabis from a chemical perspective? I mean, you and I both have looked at a lot of samples in the lab to kind of like peer under the chemical hood of these products. But what kind of diversity, from your perspective, have you seen? We'll just stick to herbal cannabis for right now.
I would guess the main diversity that we see, at least analytically, is something that's uh, it's well evidenced to cannabis aficionados, if you will,
Sure.
and
Yeah.
and that's probably its expression of different terpene types and amounts.
Hmm.
That gives rise to different aromas, and most people are fairly familiar with that. Um, there definitely is a lot of diversity um, in the cannabinoid content and
Hmm.
profile. And when I say that, I mean, um, what cannabinoids and terpenes are produced in a plant in both in maybe like a weight percent amount, but also the ratio of one to another,
Hmm.
in addition to like its mass content. And um, you and I have talked about this before. We, as analytical scientists, we talk a lot about major cannabinoids
Yeah.
and minor cannabinoids, and that can definitely extend to probably terpene expression and maybe even flavonoids and such. But I want to be clear in this discussion that if you're looking at cannabinoids and terpenes as a scientist, either medically or analytically, um, major and minor cannabinoids is a lingo that an analytical scientist would be using,
Yeah.
that just refers to its relative abundance in the plant or in the extracted material. And there's a lot of pretty provocative research that's been going on for a few years, and I know quite a bit that's going to be coming out in the
Hmm.
very near future, where because of the so-called entourage effect, a lot of therapeutic potential from cannabis as a medicine may end up stemming from what analytical scientists would be call minor cannabinoids or minor constituents.
Yeah. Yeah. And I think that's a great spot to pause and break down some of these terms that we've mentioned. So you've already, kind of pointed out that the cannabis plant, most people are familiar with cannabinoids, and you've got the like you said, the quote unquote major cannabinoids that most people think about be things like THCA, CBDA, these days CBGA, even in hemp products, hemp flower, um, and then these myriad of quote unquote minor cannabinoids, which is really becoming a misnomer these days. But things like CBC, cannabichromene, and all of these other um, slightly different cannabinoids that are structurally slightly different, like THCV,
Right.
CBDV, the diverinic
The
cannabinoids.
diverinic cannabinoids.
And for people watching or listening that maybe don't understand the difference between, like, what's the difference between THCV and THC? I mean, literally, it's just THCV has a slightly shorter little carbon chain on the molecule, but other than that, they're exactly the same. But that little carbon chain on the molecule matters a lot. Um, there's really good research coming out that shows that a lot of times the longer that tail is, the more potent that a THC type of molecule will be at a cannabinoid receptor. um, and so what look like really minor differences in chemical structure um, can result in fairly potent differences in effects. And then you have this cannabinoid fraction of the cannabis chemistry pie, and there are plenty other cannabinoids we could talk about. I mean, currently there's well over 100 that have been characterized in cannabis. I think the number is quickly approaching like 150 or something cannabinoids, and not necessarily produced by every cannabis plant, but looking at cannabis as a plant genus, it expresses um, at least 150 or more cannabinoids. Just looking at the cannabinoids, and then you mentioned terpenes as well. These kind of typically aromatic and flavor components of the plant, and you get into that class of compounds, and then now you're talking about even more, well over 200, certainly more, depending on how you think about, if you count degradation products and that sort of thing as part of the picture, it expands
Absolutely.
quite a bit more. And then you mentioned flavonoids. Do you mind explaining? Because I think flavonoids is starting to become a buzzword that's floating around in the cannabis world. People are starting to learn that it's a thing to pay attention to. But can you briefly describe like what are flavonoids? Because I think people generally have a good sense of what cannabinoids and terpenes are, but they may not have much of a background on what flavonoids are and what role they kind of serve in plants.
Sure. Flavonoids are a broad class of molecules produced by most every plant under the sun. A couple of popular ones that are known in cannabis are the cannaflavins.
Yeah.
There's cannaflavin A, B, and variants of those. Oh, if you were to look at the stick figures or like the drawings of flavonoid molecules,
Hmm.
in some sense, they resemble what you would recognize as a cannabinoid with their ring structure. Uh, why plants produce them? Flavonoids have been studied and popularized for a long time in plant medicines, but also you know other sort of industrial
Hmm.
or chemical applications as well. There's a lot of theories about why plants produce flavonoids, and
And that kind of extends to all sorts of compounds. Like we have
yes,
ideas of multitudes of roles these compounds might play in the plant, but it can be hard to nail down.
some of those functions for the plant are probably resistance against pests and
Hmm.
disease and bacteria, because a lot of flavonoid type compounds can be toxic to insects,
Mm.
bacteria, maybe even plant viruses. They're also, and I'm speaking more broadly about flavonoids in plants in general, not completely about cannabis,
Hmm.
but there's some understanding that they can provide protection against ultraviolet light. Um, there's, why do plants produce them? I don't know that we need to answer that,
Yeah.
but a lot of flavonoid compounds from a myriad of plants have worked their way into, say, dietary supplements
Yeah.
and nutraceuticals, um, because in vitro and in some context in vivo, they can provide health benefits. They can be antibacterial. A number of them can bind to various receptor types and different cell
Yeah.
types. They have uh, in once if they're ingested, once metabolized, they have broad spectrum of secondary and tertiary metabolites, which also can have biological activity. Um, there's also a strong assumption more recently that flavonoids and their metabolites in your lower GI can
Hmm.
affect gut microbiome um, uh, health and speciation.
Yeah. And what are some common examples of flavonoids that folks listening might already be familiar with? I mean, there are a couple of common ones that show up in dietary supplements and everything. I mean, one thing that I usually point out is they often provide colors to plants, and when we talk about like the antioxidant potential of like blueberries and things like that,
Absolutely.
that's a lot of times it's flavonoids that are at play there.
Oh, a really popular one would be EGCG, epigallocatechin gallate. This is
Mm.
a pretty strong constituent of green tea. It's
Yeah.
got known to have antioxidant properties. Um, I'm a little touchy about the antioxidant properties
Mm-hmm.
of flavonoids, and whether that actually has context like in our biochemistry.
Mm-hmm. Yeah.
Because a lot of those studies about antioxidant properties, they come from in vitro cell culture
Yeah.
studies where you put varying amounts of these
Done in isolation,
done
not
in
in a physiological system.
cells grown in essentially petri dishes, and then cell physiology is characterized well enough that there are all sorts of endpoints of mm, uh, mitochondrial
Yeah.
electron potential, intracellular transport. A lot of these things relate to what we would call redox biochemistry,
Mm-hmm.
and you know, at least the antioxidant potential of flavins and flavonoid compounds. It's related to their ring structure, right?
Yeah. Yeah.
They're able to absorb um, what we call colloquial free radicals and things like that. That being said, I mean, a lot of our normal homeostatic biochemistry is related specifically to free radicals
Mm-hmm.
and redox potential. And so those aren't completely bad things.
Right. Yeah, and the point that you're making about flavonoids, it extends to pretty much every chemical we talk about today, because um, I think one thing that's important to point out is that in natural products research in general, it's very common to demonstrate antioxidant potential, anti-tumor potential, that sort of thing. But in an in vitro setting where you're just exposing, these compounds to isolated cells or isolated tissues and that sort of thing. But then when you try to investigate those activities in an animal or in a human, um, things just are much more complicated. And sometimes the assumptions that we may make from the research findings of studies like that, they don't always hold up. And sometimes we even see contradictory effects in larger models and human models and stuff, just because there's so much more at play. And that's something that, and we've talked about this before, but you go into dispensaries and things and you see these charts that point out all of these constituents of cannabis, all of these cannabinoids, all of these terpenes, and they label their effects. And um, one thing that's important for people to realize is that a lot of times those charts, the data they're communicating, a lot of it is research that's done just in cell cultures and that sort of thing. That um, there's not often a meaningful way to interpret that information in a clinical way, that where you're actually going to be able to treat a medical issue. So as sort of to prime the future discussions that are about to happen, if anyone listening out here decides to go research all these chemicals and they see all this information about the different activities that they have, it's important to kind of take a step back and recognize the limitations of that information, and that we there's just really so much that we need to understand that we don't. um, can you speak to the relative concentrations in which you see some of these components? Once again, just still really focusing on cannabis flower, and then we'll spin off into other products. But what, putting the cannabinoids aside, because I think we have a pretty good understanding that generally you're going to see THCA, CBDA, CBGA, and high concentrations depending on the cultivar. Um, but looking at, like, let's say terpenes, like what are some of the kind of dominant terpenes that show up in cannabis frequently? Um, and is there anything about what you've seen when analyzing products that has kind of surprised you as far as the abundance or lack thereof of some of these components?
Yeah, if we're talking about just cannabis as an herb, typical range of concentration by weight percent for like the total terpene
Mm-hmm.
fraction.
Yeah.
Oh, it can be, 0.5, maybe up to 5%
Yeah.
by weight, in cured cannabis herb. And that would be like the total terpene fraction.
Yeah. And 5 would be like really
Very
high.
high.
Yeah, really high.
Um, and again, back to the major minor thing, just because something is minor in its weight percent doesn't necessarily mean that it's minor in its therapeutic potential.
Yeah.
And that's why there's a really, really fuzzy edge to what we call translational research, right? I
Mm-hmm.
mean, if you are culturing cells in a laboratory and at a pure cell line, and typically you would test a single compound and measure some of the physiological markers that those cells would express. That's why there's a fuzzy edge to what we call translational research,
Yeah.
in that um, and I saw this a lot during my graduate work, even just working with stuff like vitamin C and lipoic acid,
Yeah.
where you can see some pretty profound suppression of superoxide and free radical production and increase in mitochondrial membrane potential with millimolar
Mm.
levels of vitamin C. But if you look in the context of like eating vitamin C,
Mm-hmm.
and look at the type of levels that are actually available in your bloodstream, especially in peripheral tissues, micromolar or nanomolar levels are much more typical. And
Yeah.
so if you're looking at translational research with single compounds from cannabis or really any other natural product, you got to dig a little deeper to understand, like, and this relates to what we call bioavailability, right? if
Mm-hmm.
you if you eat if you drink green tea, millimolar levels of EGCG in your blood would be very, very, very high. And so looking at translational research where they're looking at molar or submolar concentrations,
Yeah.
or even high, hundreds of millimolar concentrations, possibly not uh, translatable in the
Yeah.
sense that you could never get it that high in your blood.
Yeah. I mean, even if you tried, just the basic physiological mechanisms in place to clear things out of your body, and that sort of thing, be extremely, extremely challenging to meet. That's a really, really great point. Anytime we're interpreting this preclinical research, you've got to think about bioavailability and absorption, how much of a compound as you administer it. And this links to one of the last things we'll talk about here in a little while. But, depending on how you administer a product, it's going to affect bioavailability and absorption, which is ultimately the whole point of all of that is how much of this compound, what's the concentration that's actually going to get in the blood, that's actually going to get delivered to sites of action. And even then, there's all of these ways that that's affected. And when things get in your blood, they don't just necessarily hang out freely. So they get bound by glucose and things, different things that
Absolutely.
will bind those components, possibly quote unquote inactivate them. Not a term I love, but it makes sense to a lot of people. Um,
Or the other way. I mean, binding to albumin can get compounds into more distal tissues
right.
in levels. And then there's also liver metabolism. Like you said,
Yeah.
things don't just stay in your blood until they get washed out by your kidneys. Pretty much everything that's in your blood that's not made by your body is going to be broken down by your liver. And
Yeah.
often, even the secondary metabolites that would come out of the liver have biological activity as well.
Yeah. And so, to kind of summarize all of this, what we've just presented is, the cannabis plant. We think of it as cannabinoids, terpenes, flavonoids, and other things too, stilbenoids and sterols and all sorts of other things that plants make. Um, and knowing that information, what's in a cannabis flower, is good information to have. Applying that information becomes a much more challenging task for the reasons that just highlighted just from a physiological perspective. But let's switch now to going beyond the cannabis flower, because as we know, most cannabinoid products are not actually made with flower. They're usually made with extracts for a number of reasons. So let's talk about some of the ways that cannabis extracts are produced and how the biochemistry or how the chemistry of those products changes. And then I think that's going to segue really well into coming back around to this point of methods of administration and how to think about um, how the delivery of these different chemicals in the cannabis plant will affect the body differently depending on how we ingest them or that sort of thing. So um, some of the main methods of extraction that people have probably heard of: ethanol, very common. CO2. You've got alkane extractions: butane, propane, hexane, even pentane. Pentane's usually used for crystallization, that sort of thing. And there's even some newer methods that are kind of funny, like freon's being used now as an extraction solvent, which in some ways makes sense, but it feels a little weird. um, and then there's also like mechanical methods of extraction. You can sift a cannabis flower over a screen and separate the resinous trichomes where most of these compounds we've talked about are located, and effectively concentrate them into a hash. And then there's sort of the wet method of doing that, using ice and water, cold water, to get cannabis materials, essentially to make the trichomes brittle, so that they then break off and pass through these filters more easily. Um, so in a nutshell, really quickly, that's kind of an outline of most of the extraction methods. And rosin, I guess I got to mention that too, that you can take cannabis flower or the extracted trichome heads of the cannabis plant, and you could press them under heat and pressure and make them into this kind of a taffy like substance that we call rosin. But going back to our analytical work, what are some patterns that you've noticed around the chemical constituents of these extracts versus what you see in the flower?
Most of your comment, well, to back up a little bit, the reason why extraction is an important part of producing cannabis, usable cannabis, medicinal or otherwise, is that nearly all of the terpenes and cannabinoids are what we call hydrophobic molecules. They're resinous, um, which is like they're sappy and sticky, and they do not mix with water, and that just relates to their chemical structure.
Yeah.
And so, um, almost all of those methods, I would say, other than like rosin pressing or mechanical. So all the chemical extraction methods are focused on using a chemical besides water that cannabinoids and terpenes have an affinity for and will dissolve in.
Yeah.
And so ethanol is, probably one of the most common. If you shake a cannabis flower in cold water, you know, all of those resinous fractions don't dissolve in the water.
Yeah.
And so there's no way to like get it out, so to speak, unless you're doing mechanical methods and stuff like that. And so use of hydrocarbons and alcohols and like you're saying, even uh, crazier stuff like
Tetrafluoroethane.
tetrafluoroethane and those are compounds that can essentially grab selectively these hydrophobic resin fractions of the plant, and then you can wash away the plant material, and you're left with this taffy or resin. And the different solvent types tend to favor fractions, I guess you would say,
Yeah.
of one spectrum or another. And then those solvents are all, at least to a competent chemist, relatively easy to remove
Yeah.
from that extracted matrix. And so if you shake cannabis flower in ethanol, all of the terpenes and cannabinoids and whatnot will now be dissolved in the ethanol. You can filter that out and then evaporate the ethanol. Same goes for, butane and propane
Yeah.
extractions. Those are certainly not like healthy things to ingest, but in principle, a natural product chemist can reliably remove those solvents.
Yeah.
And that being said, the use of alkanes and alcohols and organic solvents, it's in heavy use in marijuana and
Hmm.
cannabis extraction, but that's not necessarily a new thing. I mean,
Right.
in drug manufacture, um, a lot of those depend on solvent-based extraction and fractionation.
Hmm.
Um, and, to competent scientists, again, there's reliable means of removing those and testing the products to make
Right.
sure that they're
Verify the purity.
solvent-free.
Yeah.
And so, um, yeah, these days, cannabis and hemp extraction to produce crude and then also refined fractions of cannabis resins is super popular, in from recreational use all the way through to therapeutic and medical use.
Hmm.
It's definitely the way to go if you want to produce an ingestible medicine or a fraction thereof.
Yeah, and it's one thing that I like. You gave a presentation a while back about extraction, and you presented a nice sort of like picture of how to think about the chemical changes around extractions, and you kind of presented this table of like, here are these compounds and their relative abundance: cannabinoids, terpenes, flavonoids, and then all these miscellaneous components. And thinking about what slice of that spectrum are you trying to isolate, um, and each extraction method kind of approaches that a little differently. With ethanol, it's a very broad. It's a compound that has affinity for a lot of things. A lot of things have affinity for alcohol, and so it grabs a very wide spectrum, bit of that spectrum. Whereas something like butane or propane, it's going to really narrow in on those lipophilic components: cannabinoids, terpenes primarily, and flavonoids, because they're fairly fatty as well. Um, and really, really target those narrow fractions. And I've asked a lot of people, like, when you think about the term full spectrum, what does that mean to you? And while I get a wide array of answers, the most common answer that I get is when people think about full spectrum, they're thinking about the spectrum of the resin, and not
Yes.
the spectrum of the flower. Now, a lot of people don't use that term that way, and I don't like using the term full spectrum. Um, but I think it's important to point out that you may hear the term full spectrum used, and someone may only be talking about the lipophilic components of the cannabis plant and not the miscellaneous phytochemicals that aren't in the resins. And so that's just an important nuance to point out, because it can really make the conversation muddy if you don't, kind of acknowledge that. And then CO2 kind of falls in this in between ethanol and the more like strict alkane solvents. They CO2, depending on how you pressurize it, and different parameters you use, temperatures and pressure primarily. You can get it to grab not quite as wide a fraction as ethanol, but also not quite as narrow a fraction as propane and butane. A consequence of this that affects the chemistry of the product that I really want to point out is that with CO2 extraction, water becomes a big problem, because CO2 will grab water, no problem.
Absolutely.
And so if you make an extract with water, you then have stability problems, because that water is going to invite all sorts of fun microorganisms and things to start attacking away. And microorganisms also love oily things too. So
Absolutely.
having that water to feed the life process and then the oils to feed on it, it can become a kind of nasty scenario. So when folks are preparing for a CO2 extraction, a lot of times the material is totally desiccated before it goes into the extractor. The consequence of that is that if you're completely drying cannabis, most likely, well, not most likely, you are volatilizing large quantities of volatiles, primarily terpenes, out of the product before you extract it. Um, some manufacturers tackle this different ways. Some will do a terpene run on sort of fresh material, and try to kind of purge the water separately and then mix the terpenes back into their cannabinoid fraction that they run after they dry the material. But a lot of times, CO2 material, from a analytical chemist perspective, they tend to have a lower diversity of terpenoids than what you would see in propane or ethanol, and that sort of thing. The plus side is you're dealing with CO2, so it's not really um, something that you have to worry too much about from a safety perspective, worker safety perspective, other than the high pressures. You could worry about the system exploding,
Sure.
and that could be a problem. But folks sometimes like CO2 because it can be a little more environmentally friendly, and a little safer to work around compared to combustible solvents, like ethanol or propane or butane. But it seems like the consequence is, you tend to have to reintroduce a lot of terpenes if you want to kind of recapture that quote unquote full spectrum of the resins. Just heard a dog.
It's funny. I've seen this full spectrum label and even advertisement on you know what amount to pure cannabinoid distillates.
Yes, yes, and that's exactly what I wanted to talk about next was distillates.
So whether you're using uh, propane, butane, or CO2, or even ethanol, once you express all that resin out into your container, your first step is to remove that solvent, and that's really straightforward in the context of CO2 extraction.
Yeah.
It just evaporates, or in a dedicated system, it's just recondensed and back into the feed tank. Similarly, with propane and butane, that stuff is so much more volatile than the resins,
Right.
that you can just use pressurization to sort of evaporate and repump that stuff back into your feed tank. And so, invariably, what you get out of that first pass extraction is what extractors would call a crude extract, right? You've brought everything that's soluble in that extraction solvent. And along your lines of thinking, that's what I would call a full spectrum extract, right?
Mm-hmm.
That's all of the lipophilic or hydrophobic fraction. Another big constituent in quite a lot of plants, and certainly in cannabis, is waxes. And these are paraffin like compounds that are on the leaves. They provide all sorts of vapor barrier protection and stuff like that. And depending on your extraction solvent, these plant waxes end up being a big part of your first pass extraction in your crude. So the first step towards what you would call polishing of an extract would be removing of things like waxes and lipids, which at least currently, from a medicinal perspective, are undesired elements in a cannabis extract. And so a popular means of doing that is what we call winterization. You essentially dissolve that crude fraction in alcohol, Sometimes it's methanol. Typically, it's ethanol. Isopropanol, maybe sometimes. Um, you can dissolve that resin in that and use temperature, basically freezing or sub zero temperatures, where those waxes will tend to congeal together and precipitate, meaning forming like visible flakes and chunks, which are readily filtered out. Where at that temperature, the cannabinoid and terpene fractions will remain in the solvent. So that's just like a temperature gradient and filtration pass, which would give you a very clear, again, probably a quote full spectrum compound. Those winterized extracts, whether from CO2 or alkane extractions, still have this amber or golden hue, um, and still have, I would say, a lot, probably not all, of the terpene and aromatic fractions. Beyond that, it's probably a misnomer if you're polishing beyond that sort of winterized crude to call something full spectrum.
Yeah, you start getting into like a really weird discussion about like what spectrum you're talking about and how you even sort of verify your own claims. And, what we see more commonly in the hemp industry is pushing extracts all the way to these distillates. So refining beyond winterization um, to usually narrow in on the cannabinoid fraction itself. Usually with the idea that the terpenes are going to be added back in product manufacturing or whatever. And so then you've got producers that are making these products that can range wildly in purity, but that tend to have an appearance kind of like, I always call it kind of like hot glue, like cool hot glue, very semi-solid. That's really the best description I have for it. It's like hot glue. It doesn't really flow,
Mm-hmm.
um, and it's very very thick, sticky, dense.
Vitreous.
Yeah, yeah, yeah. And going back to what we see in on the analytical chemistry side, what are some unique things that you've noticed about distillates that you don't see in the other kind of we'll just call them primary extracts?
Well, this is, I don't want to get too technical, but
Yeah,
an interesting thing that we've seen over the years, and this certainly coincides with the uh, rise in acceptance and legalization of so-called adult use or recreational cannabis products, is that most of the manufacturing chemistry in cannabis resins and their um, purified or distilled products that come from that are really being driven for like commercial and adult recreational use.
yeah,
Um, though these are techniques that are, handed down and adopted again from other natural product chemistries and even pharmaceutical chemistry. Um, I think one of the main drivers in distillation of crude cannabis extracts is really to have a super high potency
yeah,
of primarily THC, but certainly CBG and CBD. So distillation is yet another form of what chemists call fractionation,
yeah,
and the idea would be if you have a de-waxed resin, it's looks similar to honey.
hmm,
It's similar consistency, maybe even a little stiffer. Um, and then those, if you're extracting high potency cannabis flower, a winterized resin, those tend to range in probably like the 30 to 60, maybe 65% THC.
Hmm.
Um, and that's analogous, I would say, to hemp extraction.
Yeah.
30 to 60% either CBG or CBD or a mix thereof. Those are pretty common in those winterized extracts. Um, and there's a lot of market for much higher and higher potency.
Hmm.
You see a lot of vape pens and other, vapable or maybe even smokable products above 60, all the way up to, at least as advertised, up to above 90 or close to
Hmm.
even 100% THC. Interestingly enough, once you get THC above about 70%, it's, at least at room temperature,
Hmm.
it's very, very stiff, hard,
Yeah.
right?
Yeah.
So I used to be baffled by seeing vape pens in the dispensary, which are labeled, 90%
Hmm.
THC. And I know as a chemist in the laboratory that 90% THC, it's nearly colorless. Um, and it certainly does not flow.
Yeah.
It definitely does not going to run or work in a vape pen. And so, um, and I don't want to, divert over to really additives, from a health
Sure. Sure. Yeah.
perspective, but most of those pens that have THC numbers, above 80%, they've either added terpenes or some other flow agent to kind of make that thing work.
Yeah.
Vape pens have little orifices and a wick, and so, like, hard crystalline material is just not going to work.
Yeah, some of that data you may be seeing a number that was representative of the distillate um, as an ingredient, and not
Yes,
as a finished
absolutely.
product. Yeah,
Yeah.
yeah. And what are one thing that we've talked about a lot that I think is really important to mention are is that through these further refinement processes, because when we're talking about distillate, we're basically talking about a step before flat out isolation. And these processes, depending on how they're done and who's the technician, running things, we're talking about all sorts of variables of pressures and heat and exposure to oxygen, all these things that can result in um, non-target impurities. Like basically you can accidentally convert your cannabinoids to other things. Sometimes cannabinoids, maybe other things. Sometimes polymers.
Absolutely.
And so, can you speak a little bit about that, of what you've seen in the lab in regards to not additives, but impurities derived from the refinement process itself?
Sure. Um, the process of distillation of I was going to say cannabinoid, but I'll say, polished or winterized
Hmm.
resins into much more high purity cannabinoid fractions involves um, um, dissolving that polished resin in typically alcohol, but sometimes other solvents, and then boiling it
Hmm.
above a column, right? And then that column, they're, depending on the technology, that, they're a foot to even
Yeah,
20
really
feet
tall. Yeah.
tall, glass column. And what happens is that at under controlled temperature and pressure, you can volatilize the cannabinoids into a gaseous phase, and then
Hmm.
they're now floating up into the column. And then you can separate them off at varying heights or pressures or even temperatures, right? And then so ideally, if you were going to distill, say, let's talk about THC resin that was 50, 60, 70% THC, you're going to run it through this distillation column, or there's other technologies, but and then it would be dripping over here onto the
Mm-hmm.
right into a flask in something that's probably above 90% pure THC. Um, Going all the way back to uh, probably the 1950s and 60s and even into the 70s, a lot of Israelis and other chemists were trying to figure out the synthetic pathways
Mm-hmm.
of how you could essentially make THC from, I want to say common lab ingredients, but readily available chemicals. And what they noticed is that cannabinoids, especially CBD, all the THC isomers and variants, their bonding structure
Mm-hmm.
and their accessory molecules that are attached to them, sometimes just methyl groups, but also carbon chains, and especially the ring structures. They're under temperatures and pressures, and I'll say also pH.
Mm-hmm.
They're
Yeah.
sensitive to opening and closing, and when molecules bonds like that open and close, um, other chemicals or molecules that are in that boiling slurry
Mm-hmm.
can react with those.
Yeah.
And um, I'll first say that today in the laboratory, we see and work with a lot of manufacturers who are doing different distillation technologies that are very sophisticated and are capable of producing really high yield, pure distillates of cannabinoids. Um, but there is still in the commercial markets less technical or
Yeah.
manufacturers
Sure. Yeah.
that are reading things on blogs and reading
Mm-hmm.
old papers and books about and approaching it from more of like a kitchen perspective.
Yeah.
Add
Mm-hmm.
this, add that, heat this, this time, this pressure. Um, and quite a few of these entities don't have the in-house analytical capabilities
Mm-hmm.
to really understand what else might be happening, as in synthetic chemistry you would call side
Yeah.
reactions, right? And so in theory, if you take CBD and heat it to the right temperature and the right solvent, with pH and pressure
Mm-hmm.
control, you can isomerize that to THC. Um, but any lack of control of any of those parameters, and you can isomerize it to maybe not delta 9 THC, but the I would say the good guys in those side reactions are THC and THC mimetics that are chemically identical to natural
Yeah.
cannabinoids. Like
Yeah.
delta 8 THC is a natural THC that is formed in some small fraction in most all plants that produce THC. But there are other and sometimes unknown or even uncharacterized um, cannabinoid like
Yeah.
molecules that can be formed in there. And I say that, these guys don't have like the in-house analytical capability to measure those or characterize them. And in a lot of ways, um, even your, you know, commercial analytical labs
Yeah.
that specialize in cannabis analytical testing don't either,
Yeah.
because most of the work that we do in a cannabis analytical lab focuses around, delivering value to cannabis manufacturers, and that relates to characterizing, what is known. And so um, it's not a simple thing for a lab, even if you're astute and accurate and precise in characterizing known cannabinoids. It's not necessarily a simple matter to go, well, what is that unknown peak that's in the
Right.
instrument? And so over the years, we have seen a vast array of what are brought into the lab as distillate products that have sure high THC content, delta 9, delta 8, but also unknown peaks, right?
Hmm.
So these, when I say peaks, I'm talking about we typically analyze cannabinoids by high pressure liquid chromatography, um, which is a means of injecting a cannabinoid sample and separating it, and then using a wavelength of light to get a reading, so to speak. And so
Hmm.
you get peaks along a line, and each of those peaks over time in the instrument correlates to a specific compound, and then that knowledge is typically validated by injecting pure analytical
Yeah.
reference standards, so you know that the time and the wavelength at which they're detected. And we've seen a myriad of distillate products that are, you know, some R&D, but some, you know, intended to go straight to market, that have these unknown, and we say unknown cannabinoid peaks, but again, these are somewhat uncharacterized. Some of
Hmm.
them are, and some of them are not.
Yeah.
And so we've seen, you know, really baffled manufacturers who bring in a winterized crude at 65% THC, run it through a really expensive distillation rig and process, and bring it to the lab, and it's clear, it looks right, and we run it on the HPLC, and now they've got 30% delta 9 THC, and you know, a sawtooth mountain range of essentially unknown or uncharacterized peaks that represent compounds that are probably side reaction byproducts, right?
Yeah.
Synthetic chemistry um, focuses on yield, which
Hmm.
means if you can work out on the chalkboard all of the chemistry reactions, ring opening and closing, and
Hmm.
aromatization and methylation and blocking, and control that. But even the best synthetic reactions give a certain yield
Right.
based on the synthetic reaction conditions. And so, without being a really sophisticated synthetic chemist,
Hmm.
um, it's nearly impossible, you know, for a basic manufacturer to understand that. And so
Yeah.
we've seen a lot of frustrated manufacturers, like I said, starting with something at 65% pure and ending up with something that's 20 or 30% pure THC.
Oh yeah. I've definitely in helping manufacturers in their in-house labs. Um I have confronted people with data like that, and understandably it is very confusing and makes absolutely no sense to them. Like, how in the world did I purify something and it's less potent? And so sometimes you receive some rather passionate responses
Yeah.
to that information.
Yeah.
People, are not stoked to.
I read it in The Alchemy of Cannabis. You cook it for 20 minutes at, you know, 3 bars of pressure, da 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 da, and like, there's no way.
Right, and you're like, well, I'm not saying there's not all sorts of other things in there. It's just a matter of if we're quantifying THC or CBD, or whatever it is. That's the way it is. And this leads into sort of like cannabinoid biotechnology, because one reason that producers, I'll say, like really well-funded, high-scaled producers, why they're looking at biosynthesis, which, for people watching or listening that aren't familiar with biosynthesis, it's basically where you're using microorganisms, bacteria or fungi, to produce a target compound. And so usually you're, examining the biosynthetic pathways in a plant or something, to understand how it's producing the compound that you want to make. And you narrow in on, okay, like, what's the basic precursor to what I need to make? What's the enzyme involved in, making that transformation happen? And then usually genetically modifying the microorganism so that it will produce that enzyme and either produce the precursors or be exposed to the precursors, and then use those enzymes to produce the target compound. And one of the main reasons that's really pushing a shift to biosynthesis is exactly what you just pointed out, that in chemical synthesis in general, you can be a super knowledgeable, talented synthetic chemist, and you're still going to have impurities, even if it's the like you're essentially getting the molecule you want, but you may not be getting it in not even on the level of isomer, but on the level of looking at the chirality of these compounds. So like you can, I hope this makes sense to people, but you could essentially synthesize both the left hand and the right hand of THC, these molecules that are the same but inverse of each other. They can't be superimposed, which is a very specific thing, and that's really hard to control. Sort of which mirror version of this molecule you're going to get. Whereas with biosynthesis, you get what that organism produces, and you get it pretty reliably, as long as conditions are maintained. And so, this is deviating a little bit from our core subject, but I did want to highlight that these problems have resulted in some very interesting work in biosynthesis, particularly using fungi as the microorganism rather than bacteria, which is it's very common to use like E. coli and stuff to manufacture chemicals. But with cannabinoids, it seems to be that companies are focusing more on fungal sources to produce those. But
Yeast.
yeah, yeast, exactly. And so to summarize what we've kind of gone through so far, and then to start to wrap this up, we talked about that the cannabis plant, the main fraction of the cannabis plant that we're really primarily concerned about when we talk about cannabis, is the resin fraction. And some of the primary chemical constituents, cannabinoids, terpenes, flavonoids, and then all sorts of other things too within the plant material itself. And then we talked about the common extraction methods and the different chemical fractions that kind of get targeted by each of those methods. And then what we just finished talking about was the refinement methods and some of the chemical impurities that can come about through those refinement methods, and some of the biotechnological ways that producers have gone about addressing those things. Now, let's talk about the kind of, not necessarily the final piece, but the other critical piece of this sort of cannabis biochemistry puzzle, which is, you've got your product, whatever it is, whether it's the cannabis flower, or you're consuming an extract, or you're consuming maybe a food that's had an extract blended into it or whatever. The other major variable is how are you consuming the product, and what is that doing? And we touched on this a little bit when we were talking about liver metabolism and all these things that happen in the body. But let's discuss some of the primary ways that cannabis products are consumed, and what those major differences look like. And I guess to start it out, I'll talk about inhalation and then pass it off to you to talk about ingestion. But when you're smoking or vaporizing something, you're relying on this gas exchange mechanism in your lungs to get compounds from that vapor or smoke into your blood. Within your lungs, you have these, I'll often joke, say they look like cauliflower, but these alveoli, these little sacs inside your lungs that are the sites of gas exchange with the blood. And so if you're inhaling a vapor or smoke that has compounds in it that can pass through the membrane there in those alveoli, then it's going to immediately reach blood vessels, and then those compounds are going to get distributed all throughout the body before they eventually get cycled, even through inhalation, eventually reach the liver and different things, and get metabolized or bound in fatty tissues in the body, and then some of it excreted. And so, when people think about cannabis, a lot of times they think about smoking or vaporizing. And with smoking or vaporizing, you get this very fast onset because of that action, that you're hijacking your lungs' ability to deliver things that are volatilized in air and to get them into the blood. And because of that, you have this onset that can be anywhere from 15 and 30 seconds to, a minute or two. Um, and in general, you're bypassing, at least immediately, a lot of metabolic pathways and stuff like that. So what you are inhaling in general is what's going to be delivered throughout the body to the brain and all of that. But how does that differ when you start talking about like edibles?
It differs in a couple of ways. Um, and you're right. You did a good explanation in that when you vaporize, either by smoking or straight vaporization, it's absorbed directly into the bloodstream. Um, that means it goes directly to your brain, and that's why
Yeah.
you feel it so quickly. When you eat something, it's absorbed through the gastrointestinal tract, and this you got to think a little bit about anatomy here.
Yeah.
Is that the blood that goes to your gut and your intestines to absorb food and nutrients and drugs or anything else that you eat, it doesn't. So you have two different sort of loops of circulation in your abdomen. And so things that are absorbed, and this protects your nervous system and your brain mostly
Right.
from the gut. Those gut veins go right into what's called the portal vein, and so it goes through your liver first, and so we call that first pass metabolism. So anything that you were to eat doesn't go directly into your bloodstream without your liver having a first crack at either absorbing, or in the case of in most cases of things that we eat, it's metabolized. Your liver produces enzymes. People are probably familiar with what's called the cytochrome P450
Yeah.
detoxification pathway. And most things that you eat, your body, if it's not a frank nutrient, is going to use a bunch of different chemical means to like break that down.
Yeah.
One of the main ways it does that is the cytochrome P450 system. It tends to kind of, especially hydrophobic compounds, it tends to break them or strip them of some of their groups so that it has a chemical charge on it, which one makes it more water soluble, and two allows later filtration and excretion. And so when you eat cannabinoids, they're passed through your liver and they're metabolized into secondary metabolites. A lot of those metabolites do retain what we would call cannabinoid receptor or receptor-like activity.
Yeah.
And so this is a common thing in pharmaceutical biochemistry and even natural product biochemistry, is that very often the compound that you eat is not the therapeutic element, if you will.
Yeah.
It's the product that comes out of a first pass liver metabolism that's the active compound. Active meaning
Right.
it's going to do what the drug or medicine is intended to do. And so when you ingest cannabis, or at least cannabis resin even, you're not circulating. So if you eat pure THC or THCA, it's you will find very little frank THC in your bloodstream. It's 11 norhydroxy tetrahydrocannabinol, which is a related compound with strong metabolic activity in the cannabinoid receptor system, but it's a different molecule than THC. And a lot of people will report vastly different high or psychoactive activity from eating cannabinoids or THC in particular, as opposed to smoking it. And that just relates to the actual molecule that's in your blood that your brain is experiencing.
Yeah, and I think I remember reading that at least roughly half of whatever THC you consume that makes it to the liver
Yeah.
will end up being converted primarily to the that main metabolite 11-OH-THC, and then all sorts of other metabolites too that can have activity in cannabinoid receptors and receptors beyond the traditional cannabinoid receptors. So it does create a weird. It creates a challenging puzzle for clinicians primarily when they're trying to think about what a product might do in the body, and if they're this ties in so well to what we were talking about earlier. That if you're studying research on a particular chemical and then you want to apply that preclinical data to clinical work, well, if someone's ingesting cannabinoids, you really need to understand the activity of the metabolites. Not necessarily, understanding the activity of the primary compound is important too, because you're going to have interplay between them all. But if you don't have an understanding of the metabolites, then you're missing a huge picture. And something I find really interesting that I like this concept a lot, that a lot of researchers now are talking about cannabinoids, orally administered cannabinoids, as essentially prodrugs, that a lot of their therapeutic activity may not be coming from THC or CBD, but rather their metabolites. And in THC, we have a pretty good sense of what those metabolites are for the most part. But there's a steep drop off. CBD, we understand generally around 4 of the couple of dozen metabolites that have been identified. And even then, what we understand about those 4 is very limited, primarily no pharmacological activity of 1 or 2 of those.
Yeah, that information comes from like forensic science.
Yeah.
That was originally designed to screen people for drug use.
Right, exactly. Yeah. And so, as far as like highlighting the state of scientific ignorance in this area, there's a ton we don't know about the metabolites of cannabinoids. And like we've mentioned here, you're talking about products that have at least dozens, if not hundreds, of cannabinoids in them. At least dozens, if not hundreds, of terpenes in them, depending on the product. And each with their own biological activity in isolation and unique activity when they're presented together, and unique activity when you consider their metabolites, and that sort of thing. And so it starts to really just highlight how complex this biochemical puzzle is around cannabis, that on multiple levels, just in looking at the flower and the complexity of the chemical constituents and trying to understand that, and then understanding the chemical complexity of the intermediate products, these extracts and everything, and then trying to understand the biochemistry of what's actually happening in the body when these things are consumed. And I think when we engage with a lot of stuff in sort of popular culture, we're given the impression that we know quite a lot about what these things are doing and what they might treat and all of that. And I hope that in people, listening to this, they maybe understand that it's really not simple, that it's fairly complex. And this is why if someone is actually trying to use cannabis medically, why it's so important to work with a knowledgeable healthcare professional, that even if they don't understand why something works, or they don't understand the mechanisms, they can at least help guide someone through the process of experimentation safely, that they can think through these different elements, like, oh, you want to take cannabinoids orally, well, what medications are you taking, and how we know that cannabinoids influence liver enzymes and change the way drugs are broken down. And CBD is often touted as the cannabinoid that does that exhibits this what's called the grapefruit effect, that tends to suppress these P450 enzymes. But all cannabinoids actually have that effect, just to varying degrees. And CBD is more potent in that effect than THC. But there's a lot we don't know about how other cannabinoids are affecting those enzymes, which is going to possibly lead to contraindications with other medications. It affects, it brings into the spotlight diet, because a lot of things we eat also influence those P450 enzymes.
The gut microbiome.
Exactly. Yeah, all of these different things. And so, to really, I guess, wrap up this discussion, the whole point of this little discussion and lesson is, you know, how does a biochemist see cannabis and cannabis products and what's going through our minds? And a lot of what we highlighted are the major things, that it is a puzzle that is so complex. You know, more so than a lot of common products on the market, like a lot of dietary supplements and stuff that are, generally uh, not too many compounds involved, and we have a pretty good sense of what they do. I've studied them clinically for quite a while and everything. Um, so I guess from our perspective here, I mean, one of the things that I want to impress is that so much about medical cannabis is still trial and error, and my hope is that by people understanding some of these issues, that maybe they'll recognize just how nuanced it can be and how important it is to work with a doctor, take careful notes, think very critically about all of these things. And some things we didn't even talk about. We talked about ingestion and inhalation, but there's also like sublingual use where you're holding things under your tongue, and then cannabinoids can pass through the membranes in your mouth and reach the blood, very similar to how it works in the lungs. It takes a little longer, but it's still pretty fast compared to ingestion. You can get onset of effects in 10 minutes, 15 minutes or so, sometimes shorter. And then there's things like suppositories, and we didn't even talk about like topical and transdermal applications. And it really just starts to get more and more more and more complex the more layers you start to stack on top of it. And I hope that folks that have tuned into this, I hope it's not overwhelming, I guess, to think about all of these things. And I don't know, some people, when I teach courses and stuff, sometimes they get a little bummed out because they're like, dang, this is like, what do we know? If
Baffling and unknowable.
Yeah.
Well, the good news, I mean, what we're talking about is, I would say, the forefront of research and understanding and maybe even developing cannabis and cannabis products as truly medicinal and therapeutic agents. But, the reason that cannabis is with us here today as humans is that its use gives us a good feeling. And that's why humans have cultivated it and bred it for years. And it's fairly well accepted to be safe in that context. And most of what we're talking about today, again, is, it's the forefront of understanding it medically and as a medicine. And contrasted to that, most of what we, our traditional paradigm of understanding medicines are single compounds. And cannabis is, it's almost never going to be that. It's a spectrum or an array of compounds. And what we know about genetics and metabolomics is that your first pass liver metabolism is not a straightforward thing. Between you and I are people of different genetic ethnicities and different world regions, and is that there's genetic tests that are available today to look at your sort of spectrum of liver metabolism enzymes. And so when you talk about ingesting cannabis, depending on your own genetics and even ethnic genetic heritage, the way that you metabolize, the speed that you metabolize, and the array of things that you metabolize, especially when they're in conjunction with other compounds that chemically and genetically manipulate that kind of stuff, it does feel baffling. That's why the scientists will work on that.
Yeah.
And it will take a while.
Right. Yeah. I like, a while back, I talked to Dr. Vincenzo De Marzo about some endocannabinoid research stuff. And it was encouraging. One of the things that he said that he really focused on is like, there's so much here to understand. It'll take multiple lifetimes, which will keep us all in business. There's plenty for scientists to explore and try to unravel and understand. And yeah, we'll never get through it in a lifetime. Well, I think that's a really good place to wrap things up. I hope that those of you that have been listening or watching, that this has been valuable to you. I hope it's provided some insights into basic chemistry of the plant, and kind of how scientists that work in labs that study these compounds, kind of how we're seeing things and thinking about things, and the questions on our minds, and what considerations you should have, and what you should think about when you're thinking about consuming a cannabis or cannabinoid product. That it is a uniquely individual thing. So be careful about listening to other people that try to tell you how something's going to affect you. Like you just pointed out, the interperson variability for a lot of these things is very substantial. So it also highlights this role of sort of N of one medicine, personalized, individualized medicine, and everything that we have to always kind of keep that in mind. And with that, if you want to learn more about the Institute of Cannabinoid Medicine and some of the stuff that we're working on, we've got all sorts of things in the works, but you can find us at tiocm.org. And if you want to study more about cannabinoid science and all this sort of stuff, Anthony and I are professors with John Patrick University, where we teach graduate courses on cannabis and cannabinoid science. So you can also look into that, and if you're ready for a deep dive, that's a great place to start. Anthony's covering a lot of the like phytochemistry, pharmacology stuff, and I'm teaching a lot on the physiology side, the endocannabinoid system, endocannabinoid dome, functions of endocannabinoids, and all of that. So we're more than happy to drown you in as much information as you're willing to take on.
Absolutely.
Yeah. Awesome. All right, everybody. Well, thanks for tuning in, and we'll catch you next time. Bye-bye.